Welcome to the Society for Developmental Biology first annual postdoctoral seminar series. We are excited to highlight the work of our outstanding postdoctoral members. Today, Dr. Christopher Chase Bolt from the École Polytechnique Fédérale de Lausanne and Dr. Nicole Edwards from the Cincinnati Children's Hospital Medical Center will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box. I have the pleasure of introducing Chase. He did his graduate work at the University of Illinois with Dr. Lisa Stubbs, examining regulatory control of the TBX18 locus in mouse urogenital development. As a postdoc now with Dr. Denis Deboul, Chase is investigating regulatory dynamics of the HOXD locus during limb and reproductive system formation. I am very much looking forward to learning more about his research today. Join me in welcoming him.
So hello, everyone. I hope you can hear me. I want to thank Camila for the introduction. It's very nice to be here. I also want to thank the Society for Developmental Biology for giving me the opportunity today to talk about our work trying to unravel the molecular origins of a limb dysmorphia called mesomelic dysplasia. This is a rare and heterogeneous group of bone disorders. Oops, 1 second, I apologize. This is a rare and heterogeneous group of bone disorders where the mid segments of the limbs are shortened and bent. You can clearly see these in the photographs of these people with this condition in the limbs. You can also see that in some individuals, this affects the legs as well, the tibia and fibia, although the changes that are present in the legs tend to be less severe. There are many families that have been affected by this, and in most cases, the mutations have been mapped to the 2q31 region of chromosome 2. The 2q31 band contains the HOXD locus, which you may know as a family of transcription factors that are very important to patterning the body during development. The HOXD locus is a 2 megabase region where the HOXD gene cluster sits at the center that you can see indicated by these purple rectangles. We know from genetic studies in mice that these genes are essential to patterning, and the limbs and mutations that disrupt the genes or the surrounding genomic environment can lead to limb malformations. One of the most interesting aspects of the HOXD locus is that the two regions flanking the cluster operate as distinct regulatory landscapes. What that means is that they contain regulatory elements indicated here as red and blue ovals that control different stages of HOXD gene expression during limb development. We refer to these regulatory landscapes as CDOM and TDOM. And in recent years, some of the human families with mesomelic dysplasia have had their 2q31 mutations mapped in higher resolution. I show you those here as the black bars. And when these mutations were mapped, they were found to vary quite a lot. In fact, some of them are deletions, others are duplications, and then there are also some inversions. The variety of these mutations then raises the, I think, very interesting question of how can such diverse mutation types and locations produce the same phenotype? We can then get some clues from a mouse model of mesomelic dysplasia on how this may work, this allele being called onulus. Onulus is an X-ray induced mutation that is semi-dominant, and it also maps to the mouse HOXD locus. Importantly, in early experiments, it was shown that mice with this mutation were expressing the HOXD13 gene ectopically in a portion of the proximal limbs that will ultimately give rise to the radius and ulna. Sometime later, the actual breakpoints of the mutation were mapped, and it was found that the onulus mutation is in fact an inversion of a large region containing the gene cluster. At the time that it was mapped, we didn't know very much about how these two regulatory landscapes operated, but it was clear that the arrangement was causing changes in HOXD gene expression in the limbs. Excuse me. This then was very suggestive that the inversion had altered the relationship between the HOXD genes and their regulatory environments. So as I had mentioned before, we know that there are two regulatory landscapes that control expression of HOXD genes during limb formation. The landscape in this bottom panel that you can see indicated by blue ovals, we call this region TDOM, and it controls the expression of HOXD gene HOXD9 to HOXD12 in the proximal limb segments. And then the landscape on the left with the red ovals drives expression of HOXD10 to HOXD13 in the distal limb domain. This means that HOXD13 is the only gene that is not expressed in the proximal limb. And this is an important clue, which then led us to the central hypothesis of this work. Is mesomelic dysplasia caused by a HOXD13 gain of function in the proximal limb? Using this question as the central hypothesis, we then broke it down into smaller testable questions. For example, do structural rearrangements cause ectopic HOXD13 expression? And if so, is the ectopic expression the primary cause of this phenotype? Then how do these arrangements produce the change in expression? And can, as a secondary effect, can we track what this ectopic expression is doing in these cells? From previous work, we know that if you simply were to invert the gene cluster itself, that this is very lethal to the embryo and is not a usable allele. On the other hand, we have the ulnulus allele, which is also no good, mostly because this is a semi-dominant allele that is also semi-lethal and semi-fertile, which makes it very difficult to do the types of studies that we wanted to do. So we reasoned that we could use a different inversion with slightly different breakpoints from the ulnulus allele that could produce the same effect, but be easier to work with. So using this rationale, we generated a new inversion that we call inversion 2, that when we produced as heterozygotes, we found that it produced no clear limb dysmorphias. But when we generated homozygous mice for this inversion, we found that, in fact, it does produce a nice, although mild, mesomelic dysplasia. When we looked at the expression of HOXD13 to see if this mutation did, in fact, produce changes in its expression, we found that by embryonic day 11.5, there was a clear domain of HOXD13 expression that was ectopic from the distal domain. And then by E12.5, this little patch of cells had moved precisely into the location of the future forearm bones. We also looked at the expression of other HOXD genes to see if they had changed in this context. And we did observe minor reduction of both HOXD9 and D10 and a minor increase of D11. So like ulnulus, these pointed specifically to the dysregulation of the HOXD13 gene as potentially being responsible for the phenotype, although we did observe some minor changes in the expression of other HOXD genes. So to ask if the ectopic HOXD13 is the primary cause for this phenotype, we turned to generating a secondary mutation in cis with the inversion. So in this case, we used a CRISPR-Cas9 approach where we cut the DNA at the 5' end of the second exon of HOXD13. And by doing this, we generated a 7 base pair deletion that causes a frame shift and premature stop codon to the N-terminal portion of the homeodomain, which is of course necessary for it to bind to the DNA. We also at the same time generated a control allele containing the same 7 base pair deletion, but on the wild type chromosome. So in this case, this is just a HOXD13 loss of function allele. Then like before, we produced homozygous mice, and what we observed is that in fact, the secondary mutation to HOXD13 does in fact completely restore the formation of the radius and ulna that you can see in this right panel here, indicated by inversion 2 with the 13 homeodomain mutation. In comparison, in our control allele, we observed no changes to the radius and ulna compared with our initial mutation in the inversion. As a quantification, what we see is that our inversion alone produces about a 20% reduction in the length of the radius and ulna. But the angular changes are most easily seen using visualized by 3D and micro CT scans. And so when you look either from the top or from the side, what you can see is how distorted the bones of the inversion allele are. And then if you go down one panel, you can see that, in fact, our rescue allele has completely restored both the angular rotation and the length of the radius and ulna. So having then established that the ectopic gain of D13 is central to the formation of the phenotype in the radius and ulna, we wanted to determine how the inversion itself is capable of producing this misexpression. So to do this, we used a chromosome conformation technique called Capture-High-C to assess how the inversion changes contacts between the HOXD13 gene and the adjacent regions of the locus. In this representation, what you see, each gray dot represents a measured contact between two regions of the DNA. So for example, a region of the genome that sits here that contacts a region of the genome that sits down here would be measured as a contact up here at the top of the triangle. And what we see in our inversion configuration is that the HOXD13 gene, represented by this purple pin, is now put into a very small and highly interactive compartment. We can see this in a different type of representation where we use a viewpoint of the HOXD gene cluster, HOXD13 again being the purple pin, and then we measure how contact frequencies change in our inversion allele versus the wild type configuration. In this case, the blue rectangles represent contacts that are more common in the inversion configuration. And what we see is that the HOXD13 gene has a very strong increase of contacts of this region of the genome here that sits between the enhancer CS65 and the next adjacent gene, that being this region down here. We then evaluated this region for the presence of some putative enhancer elements by searching for regions of the genome that have both the histone modification H3K27, and they are accessible by a TAC-seq, both of which indicate the presence of an active enhancer. And when we did this, we were able to identify 6 potential elements that are represented here as blue circles with black borders. And we took these 6 elements and we concatenated them together and we put them upstream of a LAC-Z reporter system. And then we generated transgenic mouse embryos using this system. And what we found is that in all of the embryos that stained for LAC-Z, we found some variation of this proximal limb staining pattern, including one with some distal limb staining. So from these results, we think that we can demonstrate that this inversion puts HOXD13 into contact with proximal limb enhancers. And this is what drives the expression of this gene in the ectopic patch that we see by in situ. So going forward, we can now turn to some of the downstream consequences of what HOXD13 may be doing. In order to do that, I first need to explain this relationship between 2 genes, HOXA11 and HOXD11, that are normally essential to patterning the radius and ulna. You can see both of these genes are expressed in the proximal limb domain, which will give rise to the radius and ulna. And it's been known from genetic studies that when multiple loss of function alleles for these 2 genes are combined together, it can produce dosage-dependent mesomelic dysplasia phenotype. And we know from genetic studies that HOXD13 is also necessary for the repression of HOXA11 in the wild type distal limb. But on the other hand, we don't know anything about an effect of HOXD13 on the repression of D11, and there may be none because these 2 genes normally coexist stably in the distal limb. But together these represent excellent candidates as downstream effectors for how the ectopic HOXD13 may be producing the phenotype. So to evaluate this, we first asked if we could see changes in the expression of HOXA11 and D11 in the proximal limbs of inversion 2 mutants by in situ hybridization. And we did this through a time course so that we could see if there were any minor changes in gene expression that were normally covered by saturated probe development. And what we found is that, in fact, we do see a very reproducible patch for HOX-A11 in the proximal limb that happens to correspond perfectly to the ectopic domain of HOX-D13. But on the other hand, when we looked at D11, we didn't see any significant change. On the other hand, when we look in multiple replicates of this, we do see high reproducibility for HOX-A11, but we see some variability for alterations in the expression of D11 in the same position. So on the other hand, though, in situs give a very nice bulk result of what's happening in the tissue, but what we're specifically interested in is if this repressive effect of HOX-D13 is happening inside of individual cells, and can we measure that effect on transcript levels in individual cells. So to answer this, we turn to a single cell RNA-seq approach, which I would love very much to spend a lot of time going through a lot of the interesting results that we found in here that could potentially explain a lot of what we see. But because of the time restriction, I will just focus on the main question, which is, can we detect ectopic HOX-D13 in these proximal limb cells? And if so, what effect does it have on the transcript quantities of HOX-A11 and HOX-D11? So to do this, we microdissected out this patch of cells, sort of indicated here on the diagram by the black dashed quadrangle, and performed the RNA-seq. And we first performed a clustering analysis. And when we did this, we found that in both genotypes, all clusters were detected, although we did detect some differences in the quantity of cells. But to get straight to the point, we looked for a cluster of cells that are present in our inversion mutant that normally associate with the proximal limb, but are expressing HOX-D13 only in the inversion mutants. We found that in general, the inversion 2 cells were expressing HOX-D13 at lower levels, but we detected the transcript in more cells that were associated with the proximal limb clusters. And from this, we identified 2 clusters, 1 and 6, because of their position in the same domain, in the same portion of the clustering where we normally see HOX-A11. So these 2 clusters, 1 and 6, that may be easiest to see up here in the HOX-D13 panel. And then we looked at the expression of these genes at the global level and by limb plots. And what we saw is that, in fact, there is an increase of HOX-D13 in these cells, and that we also see a minor reduction in the median expression of the HOX-A11 gene in these clusters, but we don't see any change in the expression of HOX-D11. But again, we specifically want to evaluate the expression of these genes in the same cells. So we try to step aside from the clustering analysis and use a different approach where we isolate cells from these clusters that we can detect transcripts for these different genes. And in the inversion, what we find is that there's a higher proportion of cells that are expressing HOX-D13 or HOX-D13 and HOX-A11. But at the same time, it appears that the appearance of these 2 populations appear at the expense of cells that are normally expressing only HOX-A11. On the other hand, when we look at the relationship between HOX-D13 and HOX-D11, we see a much larger increase in the proportion of cells expressing both genes. So unfortunately, I don't have time to go through this in much more detail, but our main takeaway from these experiments is that the ectopic HOX-D13 appears to directly repress inside the same cells the expression of HOX-A11, but not HOX-D11. When we evaluate this in terms of the phenotypes and how they occur during normal HOX or during a HOX-11 loss of function analysis, we don't see a reduction of these transcripts in a level that's sufficient to explain the phenotype that we observe. And finally, just to throw one last experiment in here, we wanted to assess if in this ectopic patch of the proximal limb, how the HOX-D13 protein is behaving. So to do this, we used a cut and run, which is a variant of ChIP-seq, and we performed HOXD13 cut and run on the wild type distal forelimb, as well as in the inversion to mutant proximal limbs. And when we did this, we observed that in the proximal limb, the D13 protein appears to be binding to precisely the same motif, consensus motif that it binds to in the distal limb, suggesting that the protein itself is not altering how it binds to the genome. We also performed a differential binding analysis where we compared the differential binding between HOXA11, which is normally present in the proximal limb, and HOXD13 in the distal forelimb. When we do this differential binding, we see that normally HOXD13 and A11 can bind to a large portion of sites that are the same. But HOXD13 can also bind to a large number of sites that are not normally bound by HOXA11.
1 minute.
So then we also looked at the expression of, or the binding of HOXD13 in our inversion 2 mutants. And when we did this, we found that in the proximal forelimb, it appears that the protein is binding to almost exactly the same positions that it's binding to in the distal forelimb. So from this, we think that the protein, even though its genetic and cellular context is quite different, it's still exerting a distal limb regulatory repertoire in these proximal limb cells. So just to summarize, we found that the inversion of the HOXD locus can in fact induce the expression of HOXD13 in the proximal limb. And the ectopic expression is the primary cause of the bone alterations that we observe. As a secondary effect, we think that we can exclude a role for HOX11 genes in being a downstream consequence of this gain. And then finally, when we looked at the binding pattern of the HOXD13 protein in the proximal limb, we found that it appears to be doing the same thing in the proximal limb cells as it does in the distal limb cells. So with this, I would just like to thank my PI, Denny, for being so supportive of this work, as well as my two collaborators, Benedict and Lucille. Benedict is a fantastic mouse geneticist who was responsible for most of the genetics that was presented here today, and Lucille, also a fantastic informatician and biologist, for her help in evaluating the informatics work presented here. So thank you, everyone.
Thank you for a wonderful talk, Jace. I also just want to mention that he is on the job market, and so make sure to keep an eye out for him this year. And we do have a few questions coming in. Just a reminder, you can add them to the Q&A. So we'll go ahead and start with a series of kind of 3 parts to this question. So firstly, what explains in your view the semi-dominance in the ulna-less mutant mouse? Is there a stochastic component to the expression control process? And then related, how semi-dominant penetrant is ulna-less? Yeah.
So that's a really great question, and it's one I wish that I had more time to discuss because it's one of my favorite topics. The reason that we think that this is true, and we think that it's true for the human alleles, has to do with the nature of the mutation. And that's, let me show you in a different configuration here. So in the ulna-less inversion, we think that what's happening is that the position of the breakpoints is the most relevant element here. And in this inversion, what happens is that the HOXD13 gene is now put into a position very close to what we know to be, we've characterized through multiple studies as an extremely powerful proximal limb enhancer. So we think the dominant effect comes from when you have a single allele that's directly adjacent to this very, very strong proximal limb enhancer, it induces the expression of HOXD13 in such a high level that the dosage of D13 is capable of producing the limb mesomelia from a single allele. And we think this is also true what's happening in humans.
Thank you. I should have mentioned this question is from Christopher Wright. And there's kind of a related third part, which is, could you say something more kind of touched on this, but could you say something more about the rationale behind picking your specific inversion sites in the INV2 mutation?
Yeah, so the way that we generated this allele was based on a sort of an older technique that was used before CRISPR, and this was basically you introduce a large number of LOXP sites throughout a locus, and then you can recombine those into individual LOXP sites onto the same chromosome, and then by crossing with a CRE, you can induce reconfigurations between those two LOXP sites. This was done over years of work. There were multiple LOXP sites that Denny's group inserted through the HOXD locus in order to do this kind of genetic studies. And we basically chose two LOXP sites that we thought that were in approximately the right positions that we wanted, and would give us the type of phenotype that we wanted.
Thank you. We have a question from Jessica Feldman. So first she says, great talk. And then it says, how is HOXD13 expression also targeted during evolution to create different phenotypic outcomes?
Yeah, so it's a great question, and it's been a focus of my personal work, as well as a lot of work, not just in our lab, but in lots of other groups as well. And it seems to be that this is true. There's lots of types of mutations to either the HOXD or HOXA locus that can alter the expression of these genes. It's really fascinating how both of these gene clusters have evolved with what seems to be a very similar type of regulatory strategy. That being that you have two phases of expression. HOXA is more complicated than this. But we definitely think that there's probably a large effect that comes from the evolution of how these genes are controlled, and that is essential to sort of how the modern tetrapod hands and feet especially have evolved by restricting the HOXA13 and HOXD13 expression into the most distal aspect of the developing limb.
Thank you. Our next question is from Kara Cerveni, and they ask, you mentioned that HOXD13 and HOXA, I think, bind some of the same regions in the genome. Could you say more about the different regions that HOXD13 binds, especially if they might give insight into mesomelia?
Right, so that's a great question also. So we've very recently, there was a series of publications that have come out that have shown that HOXD13 is necessary for the repression, one, of HOXA11 in the distal limb. And if you, for example, overexpress A11 into the distal limb, there was last year a publication that showed that, in fact, it's HOXD13 that's acting as a pioneer factor in the distal limb. That all of these sites that are normally bound by D13 in the distal limb are closed, and that HOXD13 is binding to those positions and opening the chromatin and potentially regulating gene expression at those binding positions. So with regard to the mesomelia, we have some various discussions going around, but unfortunately we don't have sort of a normal RNA-seq experiment on these mutants to see just how gene expression is differentially controlled in the mutant. But we kind of think that what it's doing is that you're essentially producing in the proximal limb compartment a regulatory program that is sort of pre-patterned or predefined to produce shorter bones. And as a consequence of putting D13 there, it's disrupting either the proliferation of the progenitors or their premature differentiation into the osteogenic pathway that results in the bones being so short, sort of like the bones in the hand.
Thank you. I think we do have I think we should go on to the next speaker. We do have a few more Q&A, a few more questions that Chase will answer after the seminar. So let's thank Chase again, and we'll go to our next speaker.
Thank you, everyone.
Good afternoon. It's my pleasure to introduce our next speaker, Dr. Nicole Edwards. from Cincinnati Children's Hospital Medical Center. Nicole did her PhD program at the University of Western Ontario in Canada under the mentor of Dr. Dean Bass and Dr. Andrew Walshen, studying the development of early mouse blastocysts. After that, she moved to Cincinnati for postdoc training in Dr. Aaron Zone's lab, where she studies organogenesis and disease modeling using both mouse and frog. The title for her talk today is Discovering the Developmental Mechanisms of Tracheoesophageal Defects.
All right, thank you so much for that really good introduction. And I'd like to thank the SDB for giving me the opportunity to talk about my research today. I'm interested in modeling congenital defects in animals and using developmental biology to understand new disease and new developmental mechanisms. So today I'll be talking in the context of how the trachea and the esophagus develop and the associated birth defects. And so when the trachea and the esophagus don't form properly during development, this can result in tracheoesophageal birth defects. And the most common ones that are found are esophageal atresia. So this is when the esophagus ends in a blind pouch, doesn't connect to the stomach. And this can happen with or without a tracheoesophageal fistula, which is this abnormal connection between the two structures. Some other birth defects that are observed are things like tracheal atresia or chaos, and a very severe defect, laryngotracheoesophageal cleft, where there's absolutely no separation that occurs between the esophagus and the trachea. And so you can imagine that these defects are life-threatening. Babies undergo surgery right away to correct their anatomy, but there's often a lot of long-term comorbidities associated with this. There's only a handful of gene mutations that we know causes these defects in humans. It's thought that a lot of these gene mutations are sporadic and not necessarily inherited from the parents. And so we're still really trying to understand the genetic causes of these defects. It's also challenging because actually we don't really know how the esophagus and the trachea normally develop, and this makes it challenging to study the defect. So our lab was part of a bigger research program that is dedicated to discovering the causes and improving treatments of tracheoesophageal birth defects. We're called the CLEAR Consortium. So we have a portal website that you can go to if you're interested about the project, get more information, to participate in our study, or to collaborate on the research side. And so the CLEAR Consortium is a group of 3 major projects, all involved in tracheoesophageal birth defects. We have human geneticist collaborators from Columbia University who do patient whole exome sequencing to discover new sporadic mutations in children together with their unaffected parents. We have an arm of this project looking at modeling these defects using human iPS tissue. We can generate esophageal organoids. But today I'm talking about the animal model project where in our lab, we use both the frog model Xenopus and mice in order to test new genes that are coming out of the genetic study and to discover new disease mechanisms. So for today's talk, I'll go over how we're actually able to recently define the cellular and molecular mechanisms that underlie normal tracheoesophageal morphogenesis, how we're using Xenopus to screen new patient candidate genes for disease causality, and what the role of this really new, interesting mechanism, endosomal mediated epithelial remodeling, and a new patient gene that we've discovered, and how that's implicated in tracheoesophageal defects. So it's incredibly challenging to study this process in humans because it happens so early in development, as early as 4 to 5 weeks in human development. But what we do know is what we can infer from these 3D reconstructions of the human Carnegie embryo sections. So what you see here is the developing gut endoderm tube with the future in the future trachea and esophagus here in light green and in light blue. So initially, the trachea and esophagus are a single tube in the anterior foregut. And over time, this tube splits here into the lung buds budding off, and then the nascent esophagus to eventually form these longer separate tracheoesophageal structures. So how this happens, we're not really sure. And in order to study this, we turn to our animal models. So for my research, I use both Xenopus and mouse to model tracheoesophageal development. And what we see is that compared to the human 3D reconstructions, the structure is conserved in both animal models. So early in development, the trachea and esophagus are this single foregut tube that eventually splits into two tubes. The patterning of both the digestive part of this tube and the respiratory part of this tube is also conserved between the two models. But how does this actually happen? What are the steps that occur to go from this single foregut tube to split into two separate tubes? And so we actually defined for the first time the cellular, biomechanical, and molecular mechanisms underlying how the trachea and esophagus separate from a single tube. And so here I'm just showing an overview of the steps in the Xenopus model. We first looked in Xenopus because it's highly accessible as a developmental model, and this process occurs over about 2 to 3 days, whereas in the mouse, it is much more transient. So we looked for sort of molecular mechanisms first and tested our ideas in the frogs, and then we knew what to look for subsequently in the mouse to see if it was conserved. So at about 2 days post-fertilization in the frog, the trachea and esophagus are this single foregut tube that gets patterned into 2 different domains. We have a dorsal domain that is the future esophagus here, marked by the transcription factor SOX2, and we have the ventral future respiratory or trachea domain that's marked by NKX 2.1. And so once this is all patterned, then the morphogenesis is initiated. We have this medial constriction of the mesenchyme in the epithelium to form this epithelial fusion structure, which then has to be resolved into 2 separate tubes, and the mesenchyme can come and invade the space between the 2 tubes. With time, the tubes elongate and differentiate into their 2 separate structures. And so what we know very well, actually, is how this initial patterning is set up and what signaling networks are involved in foregut patterning. And so our lab and many other labs have determined that there's this reciprocal epithelial mesenchymal signaling pathway that regulates the expression of NKX 2.1 and SOX2 in the foregut epithelium. And so briefly, the epithelium secretes hedgehog ligand, which signals to the mesenchyme, and that regulates Wnt and BMP signaling, which then creates a gradient. And this gradient is what controls whether the epithelium expresses NKX 2.1 or whether it expresses SOX2. But what happens after? What causes all these other things to initiate? And so interestingly, I'm going to show some data from the frog. We see sort of 2 distinct epithelial remodeling steps during tracheoesophageal morphogenesis. And the first thing that happens is that the epithelium, the 2 sides of the epithelial tube, constrict and fuse together to form this transient structure. And what we see in the frog is that when the 2 sides of the epithelium contact, they seem to downregulate their apical character, and that's marked by APKC, and they upregulate molecules like beta-1 integrin and E-cadherin, which we think helps promote adhesion between the 2 sides of the epithelium. Eventually, this is going to have to resolve into the 2 tubes. So what we see about a day later in the frog is that the epithelial fusion structure remodels and resolves to form 2 tubes. So here I'm going to show a video. This is a confocal Z-stack through a single frog foregut. And here, if you pay attention to the red stain, which is ECAD, you'll see sort of how the cells behave when it's time for that fusion structure to resolve. So as you progress posteriorly down this foregut, what you're going to see is that cells start to downregulate E-cadherin, round up, and then eventually incorporate it into either the esophageal epithelium or the tracheal epithelium. The basement membrane marked by laminin gets downregulated, and the FOXF1 positive mesenchyme can come in and invade the between the 2 tubes. The exact same process happens in mouse. So knowing this, we went and looked at the mouse. We saw that E10.5, we're getting this downregulation of APKC when the epithelium fuses, and this rounding up of the cells, downregulating ECAD, separating this epithelial fusion structure into 2 separate tubes. And so what we also determined in our paper was that these remodeling processes were modulated by endosomal pathways. And so cells can remodel their plasma membrane components using endocytosis or the endosomal remodeling pathways. And I'm just going to show an example of that through clathrin-mediated endocytosis. So here, there are a number of components that have to come together to create a endocytic vesicle successfully. So you have to have coat proteins like clathrin. You have to have an adapter protein that will bind the cargo. So for example, E-cadherin or a cell signaling receptor. And you have to have a protein like dynamin, which will kind of cut the membrane and form the vesicle successfully. So once internalized, the vesicle can get processed to become an early endosome. And so this is marked by RAB5. And then sort of one of 2 or 3 things can happen. If the plasma membrane components are going to be degraded, they're sent down the lysosomal pathway. Or alternatively, you can recycle these components back to another part of the plasma membrane by processing this into recycling endosomes. And these are marked by RAB11. So in this case, we're seeing the E-cadherin cargo going back to the plasma membrane and being recycled. And so interestingly, we found that this process, at least during tracheoesophageal separation, is regulated by hedgehog glee signaling. So here you can see in a control tracheoesophagus, developing tracheoesophagus, RAB11 is nicely localized along the apical membrane in the epithelial fusion structure, whereas in an embryo injected with the glutathione morpholino, we see that the epithelium and RAB11 localization is disrupted, suggesting that this process is very important for correct tracheoesophageal separation. And in fact, if you mutate different components of both the hedgehog pathway and the endosomal pathway, we see this recurring phenotype where we get a defect or a cleft in Xanthopus embryos. So in the control embryo, what we see is that the esophagus and the trachea are nicely separated, but in a GLE3 or a dynamin-2 or a RAB11 mutant, they all have this persistent epithelial fusion that isn't able to resolve at a similar developmental age to the controls, suggesting again, this is a major pathway that's involved in normal tracheoesophageal separation and could be implicated in producing tracheoesophageal defects. And so we continue to use Xanthopus to screen new sporadic mutations that come up in our human genetics part of the study. We use Xanthopus tropicalis for our genetic studies, and we're analyzing them at the F0 generation. We think this really models well that sporadic mutations that occur in children. And so introducing the CRISPR components into the embryo, we're able to genotype and phenotype the mutants only 3 days later. And so this works well because if we mutate genes that are known to cause tracheoesophageal defects in humans, we see similar defects in frogs. So for example, if you mutate GLE3 or SOX2, we get this failure to separate or this persistent cleft in the tadpoles. And so we've used this system to screen a number of genes. We have a lot more coming up in the system, but today I'll talk about one of our positive hits, ITSN1, because I think it could be implicated in this endosomal mediated mechanism, which is exciting. So the patient in our study that has a mutation in ITSN1, they have esophageal atresia and tracheoesophageal fistula. And interestingly, this gene is known to be an endocytic adapter protein. And so it's a fairly big protein. This is the structure of the human protein. It has many domains, and these have all been implicated in binding and recruiting parts of the endosome machinery to form an endocytic vesicle. So the EH domains, they've been shown to bind dynamin and synaptogenin. The DBL homology domain binds CDC42. And the plextrin homology domain binds phosphatidylinositol lipids. And all these components of the endosome machinery are very important to correctly form an endocytic vesicle. The patient has a mutation in this pH domain. It's a point mutation that results in a missense. And so based on other data and its known role in the endocytic machinery, I hypothesize that in the developing trachea and esophagus, ITSN1 acts as these red adapter proteins that recruit molecules like dynamin and actin and phosphatidylinositol lipids in order to form a proper endocytic vesicle and then move down the endosomal recycling pathway. So I first wanted to see where ITSN1 was localized in the cell. And so I mosaically expressed a GFP tagged ITSN1 in the frog, developing frog trachea and esophageal epithelium. And what I saw was that it's localized to the cytoplasm and plasma membrane, suggesting that it could be involved in these endosomal pathways. Our CRISPR targeting strategy was to model the human patient mutation by generating indels in the same domain that the patient has their mutation in. So I'm not completely replicating this patient mutation, but we're disrupting that domain that we think is disrupted in the patient. And in fact, we do see that our different models of ITSN1 loss of function and morpholino, just to confirm the phenotype, they all successfully knock down ITSN1 protein. And so what's the final phenotype? What did the trachea and esophagus look like in these different mutants? And so here you can see these are confocal 3D reconstructions of the trachea, lungs, and esophagus in the control and our different ITSN1 loss of function groups. So in the control, what we can see is that there is a small connection at the level of the larynx, which is normal between the esophagus and the trachea, but the rest of the structures have separated. Whereas in the ITSN1 mutants, what we see is that this connection between the trachea and the esophagus is significantly longer. And when we quantify this, so here in the graph, a cleft means the unseparated trachea and esophagus, the patient mutation, the loss of function CRISPR, and the morphins are all significantly have a longer cleft or a longer unseparated region compared to controls. And if we reintroduce the human ITSN1 DNA plasmid, we're able to rescue at least the morphin phenotype. And so what part of this developmental process is disrupted in order to result in that cleft phenotype? So we went back to our various steps of normal tracheoesophageal development, and I found that at this epithelial resolution step, the ITSN1 morphins were not able to downregulate E-cadherin like the controls do and resolve this structure in order to form the trachea and the esophagus. And interestingly, what I found was that there was actually quite a lot of cells kind of built up in the epithelial fusion point. So in controls, normally we really only see the structure is 2 cells thick across, whereas in the morphins, they have up to twice the amount of cells, suggesting that they're not able to resolve themselves or they're not able to migrate properly, causing this kind of buildup and this failure to resolve the structure and form 2 separate tubes. And so to conclude my talk today, what I've shown is that this new mechanism that we've discovered to be important for tracheoesophageal development, endosomal mediated remodeling, is critical for resolving this epithelial fusion point into the 2 separate tubes. And if you disrupt different pathway components of this pathway, or even hedgehog glee signaling, you result in this buildup of cells in these fusion points, and then the inability to resolve that into 2 separate tubes, causing a cleft or a tracheoesophageal defect in a frog model. And so interestingly, we think that this is an important pathway, but we found that in our list of 300 sporadic mutations that's coming out of our study, we have about half a dozen new candidates that are associated with the endosomal mediated epithelial remodeling or membrane trafficking pathways. And it'd be interesting to see if we mutate these in frogs, whether or not they reproduce the same phenotype as the ITSN1 mutants, for example. And so some future work on my end to explore a little bit more about these disease mechanisms is how exactly does ITSN1 regulate this membrane trafficking or this epithelial resolution in the trachea and the esophagus? I speculate that based on some existing data, the early endosome is not able to form properly. So you kind of get these enlarged early endosomes, and I think that disrupts the downstream process, and the cells are not able to recycle, for example, E-cadherin properly. And then as a larger question, what is actually controlling this spatiotemporal membrane trafficking or epithelial remodeling in the trachea and esophagus to ensure that you get this normal separation of the 2 tubes? So what are the upstream signals, for example, or how do the cells know when is it time to downregulate E-cadherin from the basolateral side and recycle it back to the other parts of the plasma membrane. And so I'd like to thank my lab and my mentor, Dr. Erin Zorn, our collaborators at Cincinnati Children's Hospital and at Columbia. And I just wanted to give a shout out to the National Xenopus resource, because I attended one of their workshops to learn how to do gene editing in the frogs. And I highly recommend anybody who's interested in that attend one of the workshops. So thank you, and I'll take any questions.
Great talk, Nicole. I think that's a question from Kara Cervini. So 2 questions. One, do you have any idea on how the mesenchymal epithelial signaling network comprised of hedgehog, Wnt, BMP connect specifically to the endosome pathways that you are uncovered? The second question is, does the ITSN1 loss of function mutation give a specific phenotype only in the gut, trachea, or and the esophagus?
Okay, yeah, those are really great questions. So the first one is about how do those patterning signaling networks then connect to the endosomal pathways? That's a really great question, and that's something that we're working on in the lab. Definitely, we know hedgehog is important for multiple steps of this process, not only patterning, but also the endosomal mediated epithelial remodeling. So we think there must either be some intermediate signal from the mesenchyme that then signals back to the epithelium to sort of initiate all this stuff, or there might be some non-canonical hedgehog signaling happening in the epithelium. So this is definitely something we're interested in. We're continuing to work on how to link the important signaling pathways to this new endosomal mediated epithelial remodeling. And then the second question was, is the ITSN1 phenotype specific to the gut or specific to the trachea and esophagus? No, we see some syndromic defects in the frogs. I didn't show any data today, but I have a collaboration with Helen Wilsey at UCSF. She stains frog brains, and she's interested in developmental disorders in the brain. And we actually are seeing some preliminary data suggesting that brain development is affected in these mutants.
Yeah, so that's a question from Keaton Chester, that the trachea is in frog is likely most important after metamorphosis when they transition to using their own lungs instead of gills. Do you look at the adult frogs from these morphines and CRISPRs to see if the metamorphic remodeling of the animal is able to resolve these defects, or do these defects persist?
Yeah, that's a great question. We haven't tried to take these animals out to metamorphosis to analyze them. It's tricky because the severe mutants will die before that stage. So they'll die at the feeding stage. And I've encountered that with SOX2 mutants and with ITSN1 mutants. But that's a really great point. There could be changes during metamorphosis in the esophagus and the trachea that could resolve these defects. But we haven't looked at that. So that would be interesting to look at to see post-metamorphic changes and whether or not they're able to functionally act as their organs.
There's a question from Christopher Wright. Nicole, in the ITSN mutant, are there any likely defects in the apical basal patterning, other junctions between cells, as the one tube separate into two?
Yeah, that's exactly what I want to try to investigate next. So that's a really great question. I think that there's issues with remodeling adherence junctions and tight junctions. We don't actually know what adherence junctions and tight junctions do during normal development. So that's something that I would like to characterize. We really only looked at ECAD and laminin, for example. So I would like to see if, in fact, apical basal polarity is disrupted and sort of these cell-cell connections aren't able to recycle in the ITSN1 mutants.
There's another question from Paula. Great talk. Is any connection between glee signaling and the endosome-mediated remodeling?
Yeah, so I think this was also an earlier question. There is a connection, we just don't know what the intermediate step is. So glee signaling, as far as we know, is active in the mesenchyme of the trachea and esophagus, the surrounding mesenchyme. So how that signal goes back to the epithelium to promote the endosomal remodeling, we don't know. So we're investigating potential pathways that link the two. It's also possible, well, I guess that wouldn't be non-canonical if glee is important, but yeah, we're looking for that missing black box of, you know, what connects glee to this whole endosomal mediated process.
Yeah, there's another question from August too. Nice talk. Do you think the endosome mediated remodeling may happen in other biological contexts, such as limb regeneration?
Yeah, I think that it's not unique to the trachea and the esophagus. There are actually quite a lot of membrane trafficking diseases and disorders. For example, a lot of neurological disease is a result of membrane trafficking defects. Whether or not there are other developmental processes, that would be really cool to look into. So this process is very similar to retina morphogenesis, to cleft palate morphogenesis. So it would be interesting to see if this pathway also appears in very similar developmental processes.
A question from Steven Wilkes. Nicole, great talk. Does hedgehog responses have a specific time window within the epithelia, or is it required only during their remodeling?
Yeah, that's a great question too. On our dev cell paper, we found that hedgehogs required for multiple steps of this process. So we think that there might be a window, for example, for patterning, where hedgehog glee is establishing that dorsal ventral patterning, but then it's required for this endosomal mediated remodeling and for this actually epithelial fusion as well, because some GLE2, GLE3 mouse mutants, they actually can't, those epithelium can't fuse. They just they try and then they kind of just relax. So whether or not there's control over when hedgehog is required is a great question, but definitely we know it's needed throughout the process.
There's one question from Seth Kelly, really fascinating. Do you know if there are any differences in how the trachea and the esophagus separates along the anterior-posterior axis?
Yeah, that's a great question. So we think that it happens in a posterior to anterior wave. We think most anteriorly, there might be a slightly different mechanism controlling that. And we know, for example, laryngeal morphogenesis is controlled more by cranial neural crest cells and hedgehog signaling within the cranial neural crest. So there could be some differences that account for the manifestation of these different defects, even in the humans, right? Because, I mean, you can get all these sorts of defects like esophageal atresia, whether or not you have a fistula there. So it's possible that if you disrupt, you know, somewhere along this wave, you're getting these slightly different defects occurring because of that.
Yeah, I think our last question from Jennifer White. Would a similar process apply at the other end during cloacal partitioning of the urogenital sinus and the rectum?
Yeah, that's another one of those processes like cleft palate and retina that I think is very similar to tracheoesophageal development. So it'd be interesting to look at these other midline processes to see if endosomal remodeling is important for separating the urogenital systems during development.
Yeah, I think that's all the questions for now. Thank you, Nicole.
Thank you.
Thank you, Christopher and Nicole, for the excellent talks. This seminar has been recorded and will be available online Monday. Our next speaker on February 12th, Melissa Labonte from University of Alabama at Birmingham, and Adam Isabella from Fred Hutchinson Cancer Research Center. Thank you. Have a great weekend.